Good morning, good afternoon, and good evening. Welcome back to Chapter 9, Genetics. We just finished talking about some of the terms related to Mendel's research. We talked about the initial research that he did, which is to cross different colored plants. We looked at this, and we described how you do the Punnett square to figure out what you're going to end up with. And remember that we summarized it by the F2 generation, the very last cross of the hybrids. You're going to end up with a 1 to 2 to 1 genotypic ratio. And that is one what we call homozygous dominant, purple flower, for every 2 heterozygous, which is also purple flower, for every 1 homozygous recessive, white flower. And homozygous, homo means the same, zygous means gene. So the same gene, and in this case it's the dominant gene, and for this particular trait that happens to mean purple flowers. Hetero means different, so different genes. So you get one of the dominant genes and one of the recessive. When you get different genes, which one do you show? And the answer is you show the dominant one. 2-1 homozygous recessive. Again, same gene, and this time the gene is the recessive one. In that case, the gene is the white gene. So Mendel didn't see this when he looked out across his field. What he saw was this. He saw 3 purple flowers for every 1 white flower. That's called a 3 to 1 phenotypic. Genotypic, what do the genes say? Phenotypic, what can you see? What do you see? All right. This illustrates that cross. That first generation, our true breeding parents, I cross those, purple against white, and what do their children look like? Their children all look purple because purple is the dominant trait. I cross now those against each other, and I get their children. And again, what does he see when he looks out across his field? He sees 3 purple flowers for every 1 white flower. Math, that's what he did. He did a lot of math. Calculating the statistics, calculating the math is what led him to his discoveries. The F2 generation, again, first summer, second summer, third summer. Just imagine that. It took him 3 years to get this far, just to start to be able to do the math. This shows the Punnett square, the way that you can figure out what you're going to see in that third summer. If I cross the parents, they use P for the genes. We used F. It doesn't matter. Capital P is the dominant gene for purple. Lowercase p is the recessive gene for white. Dad can either put, because remember what Dad's genes are. Dad's genes are, he's heterozygous. So he has a capital F that he could give, or a lowercase that he could get in his sperm. So in meiosis, you can't put both in there. You put one or the other. Same thing when you do an egg. Mom could either put the dominant gene in the egg, or she could put the recessive gene. And so, again, you do the Punnett square to figure out the possible combinations. And each time you have a kid, you have to roll the dice to see which combination you end up making. This is that Punnett square. So this shows the phenotypic ratio of 3 to 1, what you can see when you look out across the field, versus the genotypic ratio. What do the actual genes say these flowers should be? 1 will have both of the dominant genes and therefore be purple. 2 will have one of each gene, 1 dominant and 1 recessive, but again, if you have a mixture, you show the dominant trait, and in this case, that's purple. So 1 has both dominant genes as purple, 1 has 1 dominant and 1 recessive gene, and is therefore still purple, and only 1 will have both lowercase, both recessive genes, and only by having 2 recessive genes do you show the recessive trait. 3 to 1, 3 purples for 1 white, 1 homozygous dominant for every 2 heterozygous for every 1 homozygous recessive. 1 to 2 to 1 genotypic ratio, 3 to 1 phenotypic ratio. this is what you see when you ever had what we call a monohybrid cross. Mono means one. Hybrid means I'm mixing up true breeders. So the monohybrid cross, a monohybrid cross, you're always going to have a 3-to-1 phenotypic ratio and a 1-to-2-to-1 genotypic ratio in the third generation. The first generation is the P generation. The second generation is the initial cross. The third generation is the cross of the crosses. Mendel had 7 different traits that he had true breeding plants. And these 7 traits are listed here. He had plants that, he had true breeders for seed shape, for seed color, for pod shape, for pod color, for flower color, for flower position, and for flower height. Most of these are really easy to understand. The possible combinations for seed shape are round or wrinkled. And let's just go to the graphic. Let me show you the graphic. It's easier to see there. Okay. So here are all of the traits, the character that he had, and these are the possible versions of it. So purple versus white, flower position. This is the one that's a little bit tricky. Axial means that the flowers come off of the stem, the side of the stem. Terminal means the flowers only come off from the tip. Seed color, yellow versus green. Seed shape, inflated versus wrinkled. Pod shape, inflated versus wrinkled. Pod color, green versus yellow. And last is the height of the plant, a tall plant versus a dwarf plant. So this is 14 different true breeding plants he had to have. True breeders for this, true breeders for that. True for this, true for that, true for this, all the way down. In this particular picture, all of the ones in this first column, this is the dominant trait. And if you look at the way I listed them in the previous slides, the first trait that is listed is the dominant version of it. And then the second trait over here, this is the recessive. I only have to have one copy of the dominant gene to show the dominant trait. But the only way to show the recessive trait is to have 2 copies. And remember, you only have 2 copies of any given gene, period. One I got from my mom and one I got from my dad. So I do find it interesting, if you look really quick, pod color and pod and seed color, they both have the same variation. It's either green or yellow. It is interesting to me that yellow seeds are dominant, but green pods are dominant. And that green seeds are recessive and that yellow pods are recessive. Doesn't mean anything in the world. I just think it's kind of interesting. So he crossed 2 true breeders. We call that a monohybrid cross. And then he crossed true breeders that differed in 2 characteristics. One that was a purple flower and a tall plant against one that is white flower and recessive. And again, remember, tall is dominant, purple is dominant, white is recessive, short is recessive. He now crossed 2 of those. And so what he wanted to find out is, are all of the traits inherited all at once combined, or are they inherited separately? And we know today that we have different chromosomes, and different traits are on different chromosomes. So most of your traits are inherited separately from each other. Mendel didn't know that. He wanted to find out, because if they were all inherited together as one single intact bundle, you would not be able to separate the dominants from each other. As in, a purple dominant gene was going to be on the same structure as a tall dominant gene. And therefore, if I cross a dominant purple with a short white, I would only ever be able to get those combinations. It either has to be dominant, it would either be a tall and purple plant, or it would be short and white plant. And what he found is that is not the case. And he found that they are inherited, in fact, separately. Well, because we have different chromosomes. So this is what I'm explaining. And this is the difference in the seeds. An inflated yellow, which is the dominant characteristic in both. Yellow is dominant over green. Inflated is dominant over wrinkled. He's going to cross an inflated yellow against a wrinkled green. This is that first summer. he crosses those together, and then the second summer, all of the seeds that he collects from his garden, all of them look like that. Well, they would have to, because I have no choice but to give a big R here and a big Y. And I have no choice but to give a little r here and a little y. So if I have one copy of the dominant trait in each case, I'm going to show the dominant trait in each case. It is a large yellow pea. If they're all inherited in one bundle, that third summer this would be the only combinations you could possibly see. If it was all inherited in one bundle, if you only had one giant chromosome, you wouldn't be able to separate the genes. But what Mendel actually found that third summer is, to be fair, this mess. All right, you're not responsible for knowing this entire mess, but you are responsible for knowing a little bit of it. You can see that we do have some individuals that show one dominant trait and one recessive trait. Here's an inflated green. Inflated is the dominant trait for seed shape, but green is the recessive trait for seed color. What he found was what we call, remember that genotypic ratio here? It was 3 to 1? He found a 9 to 3 to 3 to 1 genotypic ratio. 9 to 3 to 3 to 1. The third summer, the F2 generation, that's what this is. This is the F2. That third summer, 9 seeds that he collected out of 16 are going to show the dominant trait, inflated and yellow. 3 will show one dominant trait and one recessive. These are both showing the dominant color of yellow. Or all 3 of these, rather, are showing the dominant color of yellow, but they're showing the recessive shape of wrinkled. And these 3 show the opposite. 3 show one dominant and one recessive trait. 3 show the other dominant and the other recessive trait. These are inflated in green. Those are wrinkled in yellow. And only one is going to show both recessive traits. 9 to 3 to 3 to 1. Phenotypic ratio. Oops. 9 to 3 to 3 to 1 phenotypic ratio. 9 that show both recessive traits. 3 that show one recessive trait and 9 that show both dominant traits. 3 that show one dominant and one recessive trait. 3 that show the other dominant and the other recessive trait. And only 1 that shows both recessive traits. This is a dihybrid cross. And as you can see, it's really complicated. And if you do a tri-hybrid cross, and you can do so, it's even bigger and even more complicated. But we're not going to do